Hi everyone, and greetings from Hong Kong. And so this is the first Poster Blitz session, and I hope everyone's doing well in the pandemic. Anyway, so we have a very tight schedule. We have 17 poster in this Poster Blitz session, and so the ground rule is quite simple. Please keep the time of your presentation within 3 minutes. So I don't know if we will have enough time for any questions and answers at the end, but so after each poster, that we won't be asking having a Q&A after each poster. So if we have some time at the end of this session, then we'll entertain some question. So after our time and after 3 minutes, then we'll move on to other posters. So we keep rolling. Okay, so the first presentation we have is from Ms. Tin Pang, and so without further ado, I'll hand over the stage to Ms. Pang. Thank you.
Okay, thank you. My name is Pang Ting. I'm very glad to present today. This poster gives a brief description of our study focused on prevalence of cognitive impairment and dementia in the Singapore multi-ethnic elderly cohort. A comprehensive dementia network of Singapore has been established in Singapore since 2012 to promote awareness, early detection, and timely management of CI and dementia. Hence, the awareness of dementia among Singapore elderly is believed to be improved compared to a decade ago. In this study, we estimate the prevalence of CI and dementia compared with another population-based study, which names BEAT, conducted in 2001, and that we validate whether the previously established abbreviated mental test we called AMT cutoff is applied to our cohort. Our study had two phases. In phase 1, the participants were screened by the AMT and PFQ, and those who screened positive moved on to phase 2 for further comprehensive cognitive and clinical evaluations. Here are our main findings. We recruit 3,780 elderly subjects at phase 1, and 957 of them enter into phase 2. Table 1 showed the characteristic of our participants by 3 ethnic groups. According to figure 2, the prevalence of CIND mild increased until the age of 80, while the prevalence of CIND moderate keep climbing from 1.4% to 37.3% all the time. In terms of the prevalence of dementia, which you can see the figure 3, it was consistently lower than the SPEED study before the age of 85. In the 85 years group, the prevalence was 37.5% in our study and 16.4% in the SPEED. In all, we found a 2.5% dementia prevalence using an age and race standardized adjust methods, which was higher than SPEED study. Finally, results from our RC analysis revealed that the optimal AMT cutoff is higher than SPEED and achieve better for the detection of CI. In conclusion, we found that the prevalence of dementia remained high compared to 20 years ago, stressing the continued emphasis on long-term dementia awareness, promotion, early detection, and early management. Also, with a higher cutoff on the AMT compared to 20 years ago, the test itself can still serve as a community screening tool for CI and dementia. That's all. Thank you for listening.
Thank you very much, Tin Pang. And I know that your next post is going to be you as well, right? So yes. You can start now. So please, next slide. Next poster, please.
Oh, thank you. I'd like to share with you our study focused on discriminant validity of the PFQ. That is progressive forgetfulness question. Next, please. Okay, dementia poses a serious challenge to healthcare system, especially in low and middle income countries. Although there are several screening tools in widespread use, such as MMSE and MOCA, they are not suitable for community due to time-consuming and labor-intense. So we need a briefer an assessment to apply in a primary care setting. The PFQ is a single question in screening. Our study aimed to investigate the discriminant validities of PFQ as compared to other commonly used screening tools in detecting dementia or CI in an elderly community dwelling cohort in Singapore. Our study is a two-phase community-based study. Participants were screened in phase 1 by the PFQ and AMT, who screened positive were invited to phase 2, administer the MMSE and MOCA, and validate neuropsychological battery. NCI and CND were diagnosed by the result of validated battery. Dementia was diagnosed by DSM-4 criteria. Here are our major findings. First, a total of 3,094 participants complete both the AMT and PFQ, and 918 were included in the final analysis. The PFQ showed good sensitivity and excellent NPV for ruling out healthy elderly at phase 1. Second, next slide, please. Second, at phase 2, the PFQ had very good sensitivity but poor specificity in detecting CI, high risk of dementia and dementia. Next, please. In our study, we combined the PFQ with MOCA, MMSE, and AMT to evaluate their discriminating ability on different diagnostic groups. Results show that the discriminant index didn't improve neither in a compensatory nor a conjunctive manner. Next slide, please. Finally, we examined whether diagnostic utility of 3 tools for detecting dementia differ between 2 PFQ groups. In the yes group, the 3 tools have excellent AUC. Next, please. Therefore, we recommend the PFQ can be the first step to facilitate case finding of dementia in a large population, and then conduct other screening tools to elevate the accuracy of dementia screening. Next, please. Thank you for listening.
Thank you very much, and thank you for your 2 exciting talks. So we come the 3rd presentation by March Cotto and on your poster. Okay, thank you very much. Thanks.
Hello, greetings from Finland. My name is Marge Kartau, and I am one of the researchers from Helsinki Small Vessel Disease Study. We examined the relationship between neurofilament light level, neuroimaging changes, and clinical findings in subjects with varying degrees of cerebral white matter hyperintensities. Next slide, please. The participants in our study underwent brain MRI, neuropsychological and motor performance assessments from 2016 to 2020, and 35 of them underwent additionally an analysis of neurofilament light in cerebrospinal fluid and plasma. Because cerebrospinal fluid neurofilament light levels were higher compared to plasma levels, we used the former for statistical analysis. Next slide, thank you. Our results are the following. Neurofilament light did not correlate with the volume of white matter hyperintensities. However, there were strong negative correlations between neurofilament light level and volumes of cerebral gray matter, cerebral cortex, and hippocampi. Neurofilament light was negatively associated with global cognition, executive functions, memory, and processing speed. Neurofilament light was correlated with timed up and go test results, gait speed, but not with single leg stance. This test had an upper limit of 60 seconds, which most patients easily achieved, though we think a longer time limit might make the test more informative. Plasma neurofilament light showed the same, but somewhat weaker correlations. Next slide, please. In conclusion, neurofilament light level was strongly related to global gray matter and hippocampal atrophy, but not with the severities of cerebral white matter hyperintensities. neurofilament light was consistently associated with cognitive and motor performance. Our results suggest that neurofilament light reflects frailty in the central nervous system. Thank you very much for your attention.
Thank you very much for this excellent talk, and thank you all the participants for keeping the time so well. So the next presentation, we have Robert McLachlan, so I hope I pronounced your name correctly, on your poster, please. Is Robert here? Okay. Okay, so I think we have another poster. It's from Hannah Taylor. So please go ahead, Hannah.
Hi everyone. The brain-renin-angiotensin system, the RAS, is dysregulated in AD. Next slide, please. The classical RAS pathway is overactive, while the counter-regulatory arm is defective. Neuropathology indicates that a shift in the balance of RAS is related to disease pathology in AD. Due to a lack of specific antibodies, it has been difficult to assess changes in RAS receptor expression from both arms of the pathway in AD or other common types of dementia. Our study, next slide please, our study used post-mortem frontal cortex samples from the Southwest Dementia Brain Bank in the UK, which included 50 AD, 24 mixed AD and vascular dementia, 21 VAD and 50 controls. RAS target gene expression was measured by qPCR using TAPMAN assays. We also measured standard reference genes alongside and cell type specific reference genes, which included the neuronal marker RBFOX3, which codes for NUN, the astrocytic calibrator GFAP, and the endothelial marker CD31. Relative change in gene expression was calculated using the 2 to the minus delta delta CT method. Here on the left, renin, the upstream component of the RAS, was consistently downregulated in AD. In mixed and vascular dementia, renin was lower in relation to GFAP. The renin receptor gene, ATP6AP2, was downregulated in AD and mixed groups. The classical type 1 angiotensin II receptor facilitates the main cardiovascular effects of angiotensin II and is linked to dementia pathology. In the middle graph, the expression of AGTR1 was elevated in mixed cases only. By contrast, the type 2 receptor, which counteracts the damaging effects mediated by the type 1 receptor, was upregulated in relation to the standard reference genes in AD. Finally, on the right, the AT4 or IRAP receptor is involved in memory formation and encoded by the LMPET gene. This was downregulated in AD in relation to GFAP, however upregulated in relation to standard reference genes in both mixed and vascular dementia cases. The regulatory RAS receptor MAS inhibits neurodegeneration and inflammation. MAS1 was downregulated in both AD and mixed groups. So finally, our take-home message here is that generally our data support previous observations suggesting an imbalance in RAS in Alzheimer's disease. Our novel data include lower renin and renin receptor expression and lower MAS and IRAP expression in AD, and elevated AT1 and AT2 receptor expression in AD. We also found larger changes in mixed dementia, but little change in vascular dementia, suggesting RAS signaling may be differentially altered in these dementia subtypes. Thank you for listening.
Thank you very much, Dr. Taylor. So next up, we have Dr. Güzel. Please.
Hello, everyone. My study is focused on angiotensinogen, ACE1 and ACE2 in Alzheimer's disease and vascular dementia. Renin-angiotensin system is responsible for regulation of blood pressure in human body, and ACE1, ACE2, and angiotensinogen is the component of this pathway. ACE1 is responsible for conversion of the angiotensin II from angiotensin 1 which is generated from angiotensinogen. And ACE2 is responsible for the generation of the angiotensin 1 to 7 from angiotensin 2. Moreover, some genetic studies indicate that ACE1 indipolymorphism is a genetic risk factor for Alzheimer's disease. In this study, we investigate the ACE1, ACE2, and angiotensinogen mRNA expression levels and ACE1 protein levels in frontal cortex in dementia, and examined their association with ACE1 genetic variants. For this purpose, we measured the mRNA expression levels by real-time qPCR with 3 different calibrator genes for neuronal, astrocytic, and endothelial cell expression, and ACE1 genotype determined by Taqman SNPs genotyping assay. Also, we measured the ACE1 protein levels by sandwich ELISA. When we look at my results, especially in figure A, you can see the ACE1 mRNA expression changes. We know that ACE1 is found the most in the neurons, and we found that ACE1 neuronal expression levels was significantly lower in VAD cases compared to control. Also, angiotensinogen and ACE2 mostly found in the astrocyte. And we found that in AD and mixed patients have significantly lower ACE2 expression levels compared to control groups. And in figure C, you can see the lower angiotensin expression levels in AD, VAD, and mixed cases compared to control. Next slide, please. When we look at the further analysis about ACE1 mRNA levels in frontal cortex, when we look at the figure 2, you can see the neuronal ACE1 expression levels is higher the individuals with DD genotypes compared to II genotypes who were at the high risk of the AD. Also, these ones supported by the ACE1 protein levels. When you look at the figure B, DD genotypes had higher ACE1 protein compared to II genotypes. In conclusion, alteration of the ACE1, ACE2, and angiotensinogen mRNA expression may contribute to progression of the dementia. Also, we need to further works that is underway to investigate the ACE2 and angiotensinogen protein levels and their association with ACE1 genotypes. Thank you for listening.
Thank you very much, Dr. Güzel. So next up, we have Dr. Roseborough from Canada. Please, Dr. Roseborough.
Thank you for the introduction. So today I'm going to be talking about my work looking at the role of fibrinogen, microglia activation, and extracellular vesicles. Next slide, please. So in the context of small vessel disease and aging, increases in blood-brain barrier dysfunction can involve the leakage of serum proteins such as fibrinogen, thrombin, or albumin into the brain parenchyma with potential effects on the resident cells there. Another common feature of aging and SVD is alterations to microglial signaling, involving an increase in pro-inflammatory signaling in the context of SVD. What's not well known is how extravasation of fibrinogen and microglial phenotype may influence one another. Next slide, please. To investigate this phenomenon, we exposed microglial cultures to a range of fibrinogen doses. And what you'll see on the left-hand side is that fibrinogen exposure activates pro-inflammatory signaling in microglia in a dose-dependent manner, peaking around doses of 0.1 milligram per mil, which is actually about 10 to 20-fold lower than the concentration of fibrinogen in the plasma, so it's quite low. And interestingly, if we collect the extracellular vesicles released from these cells and lyse them, we find that we can label fibrinogen within them. This suggests that EVs may carry fibrinogen as cargo. Next slide, please. This led us to ask whether or not these EVs on their own may represent pro-inflammatory signaling molecules. So we treated naive cells with extracellular vesicles collected from cells that had been pre-exposed to fibrinogen. And what you'll see on the left is that EVs on their own are capable of increasing pro-inflammatory signaling in naive cells. And on the right-hand side, if we modify either the number of EVs given, so a range of 2,000 to 12,000 per cell, or we modify the fibrinogen dose given to the donor cell, this alters the relationship between EV treatment and microglial activation. Next slide, please. So finally, in order to model subtle blood-brain barrier leakiness, where you would have a low dose of fibrinogen exposure occurring over a long period of time, we investigated whether or not repeated exposure to either fibrinogen or extracellular vesicles can result in a sustained upregulation of pro-inflammatory signaling. So on the left-hand side, you'll see repeated fibrinogen exposure, and on the right-hand side, repeated extracellular vesicle exposure. And in both cases, repeated treatment 3 hours apart results in the sustained pro-inflammatory activation of microglia cells. So in conclusion, we think that EVs may represent a novel mechanism of microglia-mediated pro-inflammatory signaling. And I'd like to thank my lab at Washington University and our funding sources for making this work possible.
Thank you very much for this very interesting talk. So next presentation, we have Dr. Bawa from Canada. Please, Dr. Bawa.
Hello, my name is Kurtleen, and today I'll be presenting my work looking at the relationships between myeloperoxidase and the cognitive and neuroimaging correlates of mild vascular cognitive impairment. So as we all know, vascular cognitive impairment is an umbrella term, and it is accompanied by vascular pathology, comorbidities, and our risk factors. It is a spectrum of disorders that can include disorders starting from the mild forms of VCI to major forms such as vascular dementia or mixed dementia. In this study, we're going to be looking at the mild forms of VCI. Next slide, please. So the disease correlates of MVCI include white matter hyperintensities, which are hyperintense regions on the T2 weighted MRI scale. We can break them into deep white matter hyperintensities or periventricular white matter hyperintensities. Next, please. And next. Patients with mild vascular cognitive impairment also have deficits in verbal memory. And in this study, we are going to be looking at verbal memory using, next please. Immediate word recall, next. Delayed word recall and word recognition. Next slide, please, thank you. So we wanna look at whether myeloperoxidase is related to the neuroimaging, so white matter hyperintensities or cognitive decline or verbal memory. So myeloperoxidase is an enzyme that is found in neutrophils, and it catalyzes the formation of reactive oxygen and nitrogen species. Previously, it has shown to play a role in vascular disorders such as cardiovascular disorders and cerebrovascular disorders, in addition to cognitive impairment. And in this study, we're going to be seeing whether there is a relationship between MPO and verbal memory or white matter hyperintensities. Next, please. So when looking at correlations, there was no correlation between MPO and verbal memory, but after controlling for BMI and education, we saw that in patients with mild vascular cognitive impairment, there was a negative association between myeloperoxidase and verbal memory, meaning that higher myeloperoxidase was associated with lower verbal memory in these patients. Next slide, please. And when looking at correlations between white matter and MPO, we saw that there was a correlation between MPO and periventricular and total white matter hyperintensity volumes, but after controlling for resting systolic blood pressure and age, the association was not significant anymore. Next slide, please. So finally, we know that cognitive decline is associated with higher risk of hospitalization and decline in activities of daily living and quality of life. And it is very important to look at new pathways and markers that might be good therapeutic targets. Our studies found that myeloperoxidase can have detrimental effects on cognition, which may be mediated by changes in white matter hyperintensity volumes in the brains of mild vascular cognitive impairment patients. And myeloperoxidase might be a novel therapeutic target for preventing the cognitive decline. With that, I would like to thank my lab and all of our collaborators and funding sources. Thank you.
Thank you, Kurtleen, for this, for your presentation. And next we have Dr. Sarait Aldrit. I'm not sure how to pronounce your name, but please start.
Yeah, thank you very much. So my project focuses on the intersection between neovascular physiology and ecotoxicology, more specifically that of urban particulate matter. All right, so next, please. So urban particulate matter has been reported for several decades to affect all sorts of human organs and tissues, as the finer size fraction is able to incorporate itself into the bloodstream through the lungs. However, in the last 2 decades, back. So in the last 2 decades, evidence of particle migration across the blood-brain barrier in young urbanites has raised concerns to the effects of long-term exposure in urban particles, mainly those derived from diesel combustion. So considering the role that astroglia plays, not just in neurovascular coupling and communication, but also as an immunoreactive cell, the hypothesis is that astroglial reactivity induced by particulate matter may drive neuroinflammation, leading to blood-brain barrier dysfunction and neurodegeneration. So next, please. So our approach consists in a transworld cell culture model, which is capable of replicating some of the physicochemical interactions key to functioning of the neurovascular unit, utilizing primary human astrocytes alongside iPSC-derived endothelial cells, has shown to improve barrier functionality based on electrical resistance, as you can see here on the right, while stimulation of astrocyte monocultures with particulate matter leads to an increased release of pro-inflammatory cytokine interleukin-6, a hallmark of astrocyte reactivity, which you can see here on the left. So the next step to follow is to characterize the effect of this reactive state on barrier stability as defined by the decrease of electrical resistance in co-culture. So as a conclusion, our hope is that by monitoring the astroglial response to particulate matter exposure and its effects on cerebrovascular integrity, a mediating role for astroglia in the pathogenesis of environment-influenced dementia will become clear. So next slide. And that's all for me. Thank you very much. I would just like to thank my lab.
Thank you very much for your talk. So the next presentation, we have Elliot Hibbs from UK, please. Please go ahead.
Thank you. Thanks. So hello, my name is Elliot. My project looks at parasite contraction in response to endothelin-1 and amyloid beta in Alzheimer's disease using a novel electrical impedance assay. Next slide, please. So cerebrovascular abnormalities, including reduced cerebral blood flow, are major contributors to cognitive decline and can occur up to 10 to 20 years before the onset or symptoms of Alzheimer's. Parasites are vascular bural cells that have a variety of functions, one of which is to regulate blood flow. And parasite tone is regulated by various chemical mediators such as endothelin-1, which is increased in Alzheimer's disease. And a recent mechanistic study has shown that capillaries are hyperconstricted in amyloid beta-rich regions in human brain slices via an endothelin-mediated mechanism. Endothelin-1 signaling has already been characterized in retinal capillary parasites, but never in human brain parasites. So the aims of this project were to investigate whether endothelin causes contraction of human brain vascular parasites and then determine the effects of amyloid on this response. Next slide, please. So to do this, we used a novel electrical impedance assay to detect morphological changes in real time that reflected contraction. It works on the principle that parasites are seeded onto a culture dish with electrodes on the bottom that pass current. The adhered cells block the current, so you could say there is a high level of impedance or resistance. As the parasite contracts, the cell rounds up, reduces in size, and the surface area in contact with the electrodes, there is less resistance. And it's this change in resistance that we use to reflect rates of contraction. Next slide, please. So using this method, we've shown that fetal-derived human parasites do contract in response to endothelin-1, and this was dose-dependent. We then compared this with adult brain-derived parasites, as these are more representative of the parasite population that you'd have in an Alzheimer's brain. And these two also contracted, but were less sensitive. Parasites express 2 receptors for endothelin, receptor A and B. The data is not shown on the slide, but by using selective antagonists, we were able to deduce that this is all mediated through receptor A. On the right-hand side, we then looked at the effects of amyloid beta on the contractile response, and we pre-exposed fetal parasites to amyloid preparations either 1 or 24 hours prior to endothelin. And we saw that while short-term exposure had no effect, longer-term exposure, particularly with amyloid beta 40, significantly dampened the contractile response. Next slide, please. So in summary, we've shown that parasites do contract in response to endothelin-1, and this is mediated by endothelin receptor A, which could prove useful in developing targeted treatments. Endothelin-mediated contraction is impaired in relation to aging, as shown by the difference in sensitivity between fetal and adult parasites. And we've shown that chronic exposure to amyloid beta, particularly A-beta 40, impairs contraction. And we plan to investigate this further to determine exactly how this occurs. Thank you very much.
Thank you very much, Elliot, for your talk. And so the next talk, we have a pre-recorded talk from Hugo Kruijff from the Netherlands. Would the controller, maybe you will play that clip, please?
One moment.
Thank you very much.
On behalf of the team in Utrecht, I would like to tell you about automatic quantification of perivascular spaces in T2-weighted images at 7 Tesla brain MRI. Perivascular spaces are believed to be involved in brain waste disposal and are associated with cerebral small vessel disease. At higher field strength, more perivascular spaces can be observed, which is challenging for manual assessment. We developed a method to automatically detect and quantify perivascular spaces. We implemented a machine learning approach that identified possible perivascular spaces in an automatically positioned region of interest in the centrum semiovale, based on signal intensities in high-resolution T2-weighted TSE scans and Franchi's Vasson's values, as you can see here in the middle picture. Next, we implemented three-dimensional perivascular spaces tracking throughout the brain in 50 persons, and quantitative measures were extracted. The results of our method show good correlations between the automatic and manual perivascular spaces count, and also for inter-observer agreement. This figure shows the distribution of perivascular spaces length throughout the brain in all of the subjects, standardized and plotted in MRI space. We observed the largest perivascular spaces lengths in the posterior regions of the centri semiovale, and shorter perivascular spaces can be observed in the anterior regions of the centrum semiovale. Furthermore, we looked at the distribution of tortuosity of perivascular spaces throughout the brain. Again, we created this three-dimensional map of all subjects in MRI space, and we observe higher perivascular spaces tortuosity in the center of the centrum semiovale, and smaller tortuosity can be observed at the periphery and in the posterior regions of the centrum semiovale. Please come visit my poster if you have any more questions, or feel free to contact me at this email address.
Okay, thank you, Hugo, if you happen to be in a Zoom now. All right, okay, so next we have Dr. Thomas Mansen from Sweden. Please.
Yes, thank you. So my name is Thomas Månsson. I work at the Department of Clinical Sciences in Malmö, Lund University. And we have investigated the connection between kidney function and cognitive function in the elderly. All data was collected from our general population cohort study, Good Aging in Skåne, or GAUS. And impaired kidney function was defined as having a glomerular filtration rate, or GFR, below 60. A cognitive test battery, including 11 cognitive tests, were used to assess cognitive function. And the domains were memory, language, complex attention, executive function, perceptual motor, metamemory, as well as global cognitive function. And first, we made a cross-sectional study with 2,431 participants. We confirmed the relationship between impaired kidney function and the cognitive domains memory, language, complex attention, executive function, and global cognitive function. We did not, however, find a relationship between kidney function and meta-memory. To our knowledge, no one has previously investigated a connection between kidney function and meta-memory. We then did a follow-up longitudinal study with 882 participants. We there found that moderately impaired kidney function measured in GFR preceded impairment in processing speed, but it did not predict dementia or mild cognitive impairment, or MCI. Processing speed is sensitive to cerebrovascular disease, suggesting that our findings could represent early vascular implications on cognition. So, in conclusion, moderately impaired kidney function preceded cognitive impairment, suggesting that overview of cardiovascular risk factors is indicated even at moderately impaired kidney function to prevent future cognitive impairment. And I would like to thank all the participants as well as all the staff of GAUS. Thank you very much.
Thank you very much for your talk. So next up, we have another talk from Sweden, from Dr. Katarina and Elström. So please.
Thank you. My name is Katarina Elström. I'm an MD and a PhD candidate at the Clinic of Geriatrics in Skåne, Sweden. The title of this abstract is Low Carotid and Diastolic Velocity is associated with white matter hyperintensities and cortical atrophy in the Swedish Good Aging in Skåne study. The relationship between hemodynamic properties and the larger arteries and cerebral small vessel disease, CSVD, is not yet fully understood. Our aim was to study the prevalence and interrelations between MRI markers of CSVD and their association to carotid artery flow parameters. And on this slide, the Good Aging thank you. The Good Aging in Skåne study is a longitudinal randomized population study of older adults ongoing since 2001. Revisits occur every third or sixth year, depending on age, and new subjects are recruited every sixth year. In conjunction with the fourth revisit, subjects were offered participation in the vessel flow and MRI sub-studies. Next slide, please. Among other examinations, we used carotid duplex to assess common carotid artery peak systolic and end diastolic velocities, and with these values, we calculated pulsatility and resistivity indexes. With the 3 Tesla MRI, markers of cerebral small vessel disease and patterns of brain atrophy were assessed by an experienced neuroradiologist using visual rating scales. Next slide, please. All subjects with peak systolic velocity indicating carotid stenosis were excluded from analysis. Mild grades of white matter hyperintensities and medial temporal lobe atrophy were considered non-pathological. Pathologies were found in 80% of subjects, and a quarter of subjects presented with only a single kind of pathology. In regression models adjusted for age and sex, we investigated the effect of carotid artery flow parameters on MRI markers of CSVD. Low end diastolic velocity was associated with the presence of white matter hyperintensities, global cortical atrophy, parietal atrophy, and a number of MRI pathologies. There were no associations between CSVD and peak systolic velocity. In conclusion, low end diastolic velocity has been proposed as a marker of arteriosclerosis. And in this cross-sectional design, it's not clear whether the low end diastolic velocity reflects a higher downstream resistance or can be attributed to arterial stiffness, in turn affecting the brain microcirculation. Thank you for your attention.
Thank you very much for your talk. And I want to thank all the participants again for keeping the time so well. So next up, we have Michelle Jansen on her talk. Please go ahead, Michelle.
Hello, everyone. It's really good to be here today. My name is Michelle, and today I would like to talk with you about our study, which aims to characterize the associations of total cerebral small vessel disease burden with brain structure, trajectories of vascular risk factors, and cognitive functions from mid to late life. Next slide, please. For the present study, we included a total of 623 subjects from the Whitehall 2 imaging sub-study, which is part of a larger parent study, namely the Whitehall 2 study. The Whitehall 2 study has acquired data on vascular risk factors and cognitive functions between phase 3 and phase 12, spanning up to 25 years in total. MRI data was only acquired at the imaging sub-study of the Whitehall 2 study, which took place at around phase 12. We calculated total small vessel disease burden scores using the STRIVE criteria, and we used FSL to acquire measures of gray matter density and white matter microstructure. Subsequently, we used general linear models to investigate the association between these imaging parameters and the small vessel disease burden scores, and we investigated whether these associations were additionally modified by cognitive status as defined as having a MoCA score of under or above 26. And we also used linear mixed effect models in order to investigate the associations of late-life small vessel disease burden with 25 years retrospective trajectories of the vascular risk factors and cognitive functions. Next slide, please. With regards to the concurrent brain structural associations, we found that higher levels of small vessel disease burden were associated with reduced gray matter density in almost 10% of the total gray matter volume. As you can see, it was quite widespread, lower fractional anisotropy, and higher levels of mean diffusivity, radial and axial diffusivity. And these latter associations were especially pronounced in individuals with MoCA scores of under 26. Next slide, please. With regards to the retrospective trajectories, we found that late-life small vessel disease burden, that those with the highest levels of small vessel disease burden demonstrated already elevated levels of mean arterial pressure at baseline, which also proceeded throughout the course. With regards to the cognitive functioning, we found that these individuals with the highest levels of burden also demonstrated relatively steeper decline rates in phonemic fluency and verbal reasoning. Next slide, please. Altogether, the associations that we found of late-life small vessel disease burden with several concurrent brain structural alterations with both gray and white matter, as well as increased mean arterial pressure we found at baseline and steeper cognitive decline rates further emphasize the importance of managing midlife vascular health to maintain brain structure and function in late life. Thank you, and please look up our poster and preprint.
Thank you, Michelle, for this very interesting study. So next talk, we have Elizabeth Kliam. Please, Elizabeth, let's go ahead.
Hi, everyone. My name is Elizabeth, and I'm a second-year PhD student in the Vascular Disease Research Group at NTNU Norway. I'm very happy to present our recent study on self-reported daytime sleep, emotional symptoms, and cognitive function after ischemic stroke today. Next slide, please. And next again. And next. Thank you. We know that after stroke, symptoms such as depression or anxiety, cognitive decline, and increased daytime sleep are common. However, the relationship between these symptoms still remains unclear. So we aim to study if self-reported memory and concentration and anxiety and depression 3 months after hospital discharge are associated with increased self-reported daytime sleep at 1 year. In addition, we also aim to study if increased self-reported daytime sleep at 3 months is associated with emotional symptoms and cognitive decline at 1 year. Next slide, please. To study these relationships, we included ischemic stroke patients without recorded previous history of dementia or depression. We assessed self-reported symptoms via postal surveys. More specifically, symptoms of anxiety and depression were assessed with the hospital anxiety and depression scale, and self-reported increase in daytime sleep and decline in concentration and memory were each assessed using dichotomous items. In addition, we also used data from patients' medical journals. Next, please. Our sample consisted of 140 patients with a mean age of 73.2 years and 57.1% were male. Strokes were rather mild with an NIH stroke scale score of 3.6 within 24 hours of stroke, and mini-mental state examination score was 25.2 out of 30. For our analysis, we used multiple linear and binary logistic regressions controlling for age, sex, and stroke severity, and we used Bonferroni correction to correct for multiple analysis. Next. We found that self-reported decline in memory and concentration, as well as higher HOTS-A and HOTS-D scores at 3 months, were significantly associated with self-reported increased daytime sleep at 1 year. Next, please. However, self-reported daytime sleep at 3 months was not significantly associated with self-reported cognitive function or emotional symptoms at 1 year. Next slide, please. In conclusion, this may indicate that patients with self-reported higher levels of emotional symptoms and decline in cognitive function 3 months after stroke may be at a higher risk for increased daytime sleep at 1 year. We suggest that interventions targeting emotional symptoms and cognitive decline may prevent excessive daytime sleep and may thereby secure patients' engagement in rehabilitation and successful recovery. If you want to find out more about our results and methods, please visit our poster number 35. And finally, I would like to thank my supervisor and my research group and you for your attention. Thank you.
Thank you very much, Elizabeth. And the next presentation, we have Elise Jastad. Please go ahead.
Thank you so much. I'm Elise Jastad, and I'm also a PhD student from the Vascular Diseases Research Group at NTNU. I'm presenting the findings from our study exploring possible social cognition impairments in stroke patients. Next slide, please. So after the inclusion of impairments to social cognition as a diagnostic domain of post-stroke neurocognitive disorder in the DSM-5, we wanted to examine the link between this and general cognitive function in a stroke population. Tests of social cognition were therefore added to a large ongoing study, the Norwegian Cognitive Impairment After Stroke Study, or NORCOST, which is a longitudinal cohort study of acute stroke spanning a large geographical area of Norway. 29 patients agreed to a longer follow-up 3 years post-stroke, including a larger neuropsychological test battery. 2 different aspects of social cognition were measured using the hinting task and the pictures of facial effect, where the hinting task is a measure of theory of mind, or the ability to consider the mental state of another, and the picture of facial effect is a measure of emotion recognition, using the Ekman stimulus set with an array of different facial expressions. This type of testing of social cognition has not previously been used on stroke patients in Norway. And finally, we used the Mini-Mental State Examination, the MMSE, as a measure of general cognitive function. Next slide, please. On average, patients correctly identified 74.9% of the facial expressions, while for the hinting task, the mean score was 18.9 out of 20. The distribution of score on the pictures of facial effect is shown on the right. Linear regression analysis showed that a lower score on the MMSE predicted a lower percentage of correctly identified emotions on the picture of facial effect test, while there was no association between the MMSE and the hinting task. As indicated by the average score on this task, the variation was limited, which may indicate a ceiling effect. Next slide, please. In conclusion, the results supported the initial hypothesis that there would be an association between general cognitive function and social cognition, and it shows that a relatively simple task like the picture of facial effect could be used for examining these impairments. We plan on expanding this further using a larger sample and a control group. The results also highlight the importance of bringing awareness of the possibility of social cognition impairments to a clinical setting, as this cognitive domain may influence the patient's functions in all situations involving other people. Any further details on the study and our methods may be found in our poster, and thank you all for your time and attention.
Thank you very much, Elise. And here comes to our last poster in this session. It's from Amy Jolie from UK. Amy, please go ahead.
Hi everyone, thanks for the opportunity to talk today. So my name is Amy and I'm a first year PhD student at the Stroke Research Group in Cambridge. And I'm going to talk today about our research looking at the prevalence and predictors for vascular cognitive impairment, or VCI, in CADASIL. So CADASIL is the most common inherited form of stroke and is associated with recurrent stroke and early onset dementia. And we established cognition using the brief memory and executive test and the Montreal cognitive assessment in 176 genetically confirmed CADASIL patients and 265 healthy controls. And VCI was determined as per the standard predefined cutoffs for each assessment. We then tested the association of VCI with clinical risk factors such as stroke and migraine with aura, the location of the mutation, and MRI parameters. When defined by the BMAT, VCI was present in 39.8% of the CADASIL patients and 10.2% of the controls. And when it was defined by the MOCA, it was present in 48.9% of the CADASIL patients and 21.1% of controls. And as can be seen on the graph, CADASIL patients performed worse than controls in all of the domains of the BMAT. And this difference was significant. And we can also see here that CADASIL patients with stroke performed the worst overall. And on logistic regression, stroke was the only predictor of both VCI as defined on the MOCA and on the BMAT. And stroke was associated with just over twice the risk of VCI defined on both these measures. And in addition, LeCun count was also associated with just over 1.5 times of the risk of VCI as defined by the BMAT. So in conclusion, VCI is present in 40 to 50% of CADASIL patients with a mean age of 50. Reductions were seen across all cognitive domains, and stroke and LeCun count were independent predictors of VCI. So thank you very much.
Thank you very much, Amy. So thank you again for all the presenters. So here comes, we have, I think, about 7 to 8 minutes of Q&A session. So you can just unmute yourself and ask the question. Please identify yourself and to tell us the name of the speaker that your question is addressed to. Okay, so the floor is open. Thank you. Or you can also type your question in the chat. Okay, maybe I can start a question first. So this question is for Ms. Tian Pan, and for your first talk, I noticed that you have used the AMT actually for the classification or to define dementia in this study. But I understand they probably you use it because it's short and it's very easy to administer. But do you think you would be like you know, be sacrificing some of the sensitivity because it is too simple, and a lot of people, especially those with more, you know, a brighter background or higher education, will actually be missed using this, you know, simple AMT test? Thank you.
Sorry, I can't catch your point. Can you repeat again?
Oh yes. So I saw a study use the AMT to classify dementia in your epidemiological study, right?
Yeah.
So as we know, the AMT is very brief, it is very convenient, but it is also not very sensitive. So what do you think about, you know, how about, you know, missing cases, especially those with higher education or a more sophisticated, you know, intellectual background?
We recommend to use AMT and PFQ in a large community screenings. So we thought that using that abbreviated task can ruling out the elderly people in a large community screening. So we always recommend use the PFQ or AMT at the first step for ruling out the elderly people. Thank you.
Mm-hmm. So it's a stepwise process.
Yes.
Okay, so we have a question from Ellen. So in what circumstances are you recommending screening for dementia?
We recommend at the communities larger population screening, or some in the such as in the primary care setting.
Okay, thank you very much.
Thank you.
Thank you. So anyone has any question to any of the speakers? So I have another question for Elise. So I think social cognition is really an under-researched area, although it has been mentioned in the DSM-5 for as in the diagnostic criteria for neurocognitive disorder in the DSM-5, which was published in 2013. And I think it is like there should be a lot more research in this area. So I've seen in your slides that you were correlating these the test, the social cognition test with MMSE, but actually they tap like really different cognitive processes. Like the MMSE is more about the more conventional cognitive domains, while the other tests that you have included is more social cognitive oriented. So may I know if you have also other like more formal or other like assessment of social cognition that you will compare your scales against with in this population, or and what are what is actually the reason for correlating the social cognitive test with MMSE? Thank you.
Thank you for your question. We wanted to look at this specific correlation precisely because of regression, because of the connection that has been posed by the DSM-5, that there should be limitations to this domain in patients who have post-stroke neurocognitive disorder. We do have some other data on this population, which is currently still under processing. So I cannot tell you what this data will lead to. We have some other cognitive measures, and we also have some measures of emotions and also frontal functions, but it's still not completely done with the analysis, so I cannot tell you if there is anything there.
Okay, thank you. So is there a sensitive neurocognitive, social cognitive test that you can recommend?
From our results, we have only really tried out hinting task and the pictures of facial effect, and we have definitely had most success with the pictures of facial effect. So that is, I think, the best I can.
Because nowadays most of these social cognition tests are borrowed from the autism area, so they are more commonly used in the assessment of people with autism. So I just wonder if there is like any like good test for use for the adult or elderly population. Anyway, thank you very much. Yes.
Thank you so much.
Thank you. So anyone has any questions for any speaker? All right, so I certainly enjoyed this session very much, and thanks again for all the participants and all the audience here. So I think we can close this session, and next I think we have a break. So yep, so see you around in Vascular. Please enjoy.